Episode 2, Before Mechanisms. A bacterial cell divides every 20 minutes. In that brief window, it can mutate, it can acquire resistant genes from neighbors, it can pump out antibiotics, or produce enzymes that destroy drugs. Evolution operates in real time. I'm Dr. Anthony Ray Anaweokhai. On this channel, we decode global health through evidence-based analysis, one crisis, one solution at a time. This content is educational and is based on WHO 2024 to 2025 data and CDC reports. It's not medical advice. For antimicrobial prescribing or stewardship implementation, consult qualified healthcare professionals and infectious disease specialists. To combat antimicrobial resistance effectively, we must first understand how bacteria develop their defenses. Now, the mechanisms are neither mysterious nor random. They follow predictable patterns rooted in evolutionary biology, accelerated by our misuse of antimicrobials. And the World Health Organization identifies 4 primary mechanisms through which bacteria acquire resistance. Each mechanism presents a distinct survival strategy honed over millions of bacterial generations. Together, they create a formidable arsenal that threatens to render our most powerful antibiotics obsolete. What makes this mechanism particularly dangerous is the speed. While human evolution operates over millennia, bacterial evolution occurs in real time. A single bacterial colony can contain more individuals than the entire human population. Each division, occurring every 20 to 30 minutes, represents an opportunity for adaptation. The data from the Gram Project 2024 reveals that multi-drug resistant pathogens now employ multiple mechanisms simultaneously. Escherichia coli strains isolated from bloodstream infections demonstrate resistance through 3 or 4 mechanisms concurrently. Klebsiella pneumoniae shows similar patterns. These convergence transform treatable infections into clinical emergencies. Understanding these mechanisms is not merely academic. It informs clinical decision-making, it guides antimicrobial stewardship protocols, and it directs research priorities. The CDC's 2021 Antibiotic Resistance Threats Report emphasizes that mechanism-based surveillance now drives containment strategies across healthcare systems. As the ancient philosopher Lao Tzu observed, knowing others is intelligence. Knowing yourself is true wisdom. In the context of antimicrobial resistance, knowing our microbial adversaries, the strategies, their vulnerabilities, their evolutionary pressures, constitutes the foundation of effective response. The first mechanism is spontaneous gene mutation. Every time a bacterial cell replicates its DNA, error occurs. Most mutations prove harmful or neutral, but occasionally a random change in genetic sequence confers survival advantage in the presence of antibiotics. Consider the mathematics. A single bacterial cell dividing every 20 minutes produces over 1 million descendants within 7 hours. Each division creates opportunity for mutation. Under antibiotic pressure, susceptible bacteria die while resistant mutants survive and proliferate. Natural selections occur at antimicrobial speed. The CDC documents this phenomenon extensively in hospital settings. Methicillin-resistant Staphylococcus aureus emerged through mutations in genes encoding penicillin-binding proteins. These proteins normally serve as antibiotic targets. Mutated versions maintain their cellular function while evading drug binding. Fluoroquinolone resistance in gram-negative bacteria follows similar patterns. Point mutations in genes encoding DNA gyrase and topoisomerase enzymes, the targets of fluoroquinolone antibiotics, reduce drug affinity while preserving enzymatic activity. The bacterium survives. antibiotic fails. Now, what amplifies these mechanisms impact is selective pressure. Inappropriate antibiotic prescribing creates environments where resistant mutants thrive. The WHO's 2024 surveillance data shows that regions with highest antibiotic consumption demonstrate highest mutation-driven resistance rates. The correlation is unmistakable. Mutation-driven resistance develops gradually. This offers opportunities for intervention. Antimicrobial stewardship, which means using the right drug at the right dose for the right duration, minimizes selective pressure. Diagnostic stewardship, confirming bacterial infection before prescribing, prevents unnecessary exposure. These strategies slow mutation-based resistance development. The challenge lies in implementation. Even optimal stewardship cannot prevent spontaneous mutations entirely. Evolution remains inevitable. Our goal is not elimination but delay, buying time for new therapeutic development and preserving existing antibiotics. Now, the second mechanism is horizontal gene transfer, and it operates with alarming efficiency. Unlike mutation, which requires time and selection, horizontal gene transfer allows bacteria to acquire complete resistant genes from neighboring cells within minutes. Now, three mechanisms facilitate horizontal gene transfer. Conjugation involves direct cell-to-cell contact through pilot structures, transferring plasmids containing multiple resistant genes. Transformation occurs when bacteria absorb DNA fragments from dead neighboring cells. Transduction employs viral vectors called bacteriophages to move genetic material between bacterial hosts. The public health implications prove profound. The Lancet 2024 analysis demonstrates that carbapenem resistance in Enterobacteriaceae spreads primarily through plasmid-mediated transfer. A single plasmid may carry genes conferring resistance to multiple antibiotic classes, beta-lactams, aminoglycosides, fluoroquinolones. This creates multi-drug resistant superbugs from previously susceptible strains. The environmental dimension compounds the challenge. Horizontal gene transfer occurs not only in clinical settings, but in soil, water, and agricultural environments. Resistant genes originating in livestock production facilities transfer to human pathogens through environmental bacteria serving as intermediary hosts. The US Centers for Disease Control's One Health surveillance confirms this pathway contributes significantly to resistance dissemination. Hospital wastewater presents a particularly concerning reservoir. High antibiotic concentrations, dense bacterial populations, and constant selective pressure creates ideal conditions for gene transfer. Studies from the WHO Global Antimicrobial Resistance Surveillance System document resistance gene exchange in hospital sewage systems with subsequent environmental contamination. The speed of horizontal gene transfer demands urgent containment strategies. Now, unlike mutation-based resistance requiring generations to spread, transferred resistance can cross species barriers in hours. This necessitates infection prevention and control measures that limit bacterial transmission, regardless of resistance status. As Aristotle wrote, the whole is greater than the sum of its parts. In bacterial populations, collective gene sharing mechanisms create resistance storage exceeding what individual mutation could achieve. Now, the third mechanism involves efflux pumps. Protein structures embedded in bacterial cell membranes that actively expel antibiotics from cells. Think of them as molecular bouncers, ejecting drugs before they even reach therapeutic concentrations. Efflux pumps exist naturally in most bacteria, evolved to remove toxic compounds from cells. Now, under antibiotic pressure, bacteria overexpress these pumps through genetic mutation or regulatory changes. Increased pump activity reduces intracellular drug concentration below lethal thresholds. The versatility of efflux pumps poses particular Challenges. Many pumps demonstrate broad substrate specificity, expelling multiple antibiotic classes simultaneously. Pseudomonas aeruginosa produces efflux pumps that eject beta-lactams, fluoroquinolones, and tetracyclines concurrently. This creates multi-drug resistance through a single mechanism. The WHO's 2024 antimicrobial resistance surveillance identifies efflux-mediated resistance as increasingly prevalent in gram-negative pathogens. Hospital-acquired infections caused by Acinetobacter baumannii and Pseudomonas aeruginosa frequently involve efflux pump overexpression, complicating treatment options and extending hospital stays. Research efforts focus on efflux pump inhibitors, main compounds that block pump function, restoring antibiotic susceptibility. Early-stage clinical trials show promise. However, development challenges persist. Pumps serve legitimate cellular functions. Complete inhibition may prove toxic to human cells or create unintended bacterial stress responses. Current clinical approaches emphasize combination therapy. Pairing antibiotics with different molecular structures can overwhelm pump capacity or exploit alternative cellular targets. This strategy, guided by susceptibility testing, improves treatment outcomes in efflux-mediated resistant infections. The fourth mechanism now is perhaps most direct. Bacteria produce enzymes that chemically destroy antibiotics. Beta-lactamases, the most clinically significant examples, cleave the beta-lactam ring essential for antibiotic activity. Once broken, the drug becomes therapeutically useless. The diversity of these enzymes creates ongoing clinical challenges. Researchers have identified over 1,000 distinct beta-lactamase variants. Each variant demonstrates unique substrate specificities and inhibitor sensitivities. Extended spectrum beta-lactamases hydrolyze third-generation cephalosporins. Carbapenem destroy even last resort carbapenem antibiotics. The CDC's 2021 antibiotic resistance threat report classifies carbapenem resistant Enterobacteriaceae as an urgent threat, largely due to carbapenemase production. These infections carry mortality rates approaching 50%. Treatment options become severely limited. Clinicians resort to older, more toxic antibiotics like colistin, itself driving nephrotoxicity and new resistance patterns. The pharmaceutical industry has responded with beta-lactamase inhibitors, compounds that inactivate these enzymes, preserving antibiotic effectiveness. Combinations like amoxicillin clavulanate and piperacillin tazobactam represents this strategy. Newer inhibitors like avibactam expand the spectrum of neutralized enzymes. However, bacteria continue evolving. Recent surveillance identifies metallo-beta-lactamases, zinc-dependent enzymes resistant to current available inhibitors. This ongoing evolutionary arms race demands continued research investment and novel therapeutic approaches. The ancient strategist Sun Tzu taught that the supreme art of war is to subdue the enemy without fighting. In antimicrobial resistance, preventing enzyme production through reduced antibiotic pressure represents our most effective long-term strategy. Healthcare professionals, which resistant mechanism most frequently complicates your clinical practice? Share your experiences in the comments. 4 mechanisms: mutation, horizontal gene transfer, efflux pumps, enzymatic destruction. Each operates through distinct molecular pathways. Together, they create the antimicrobial resistant crisis confronting global health systems. Now, understanding these mechanisms informs clinical decision making, guides research priorities, and shapes policy interventions. The evidence is clear. Bacteria evolved continuously. Our response was proved equally adaptive. Next episode, agriculture's 70% problem. The largest consumer of antibiotics isn't even hospitals, it's farms. You won't want to miss it. Subscribe to Global Health Decoded, Instagram, TikTok, Facebook, LinkedIn at Global Health Decoded. Next, agriculture's 70% problem. Spotted the DNA helix in our logo? Comment your observation.